For 40 years, scientists tried to read the genetic code locked inside Egyptian mummies. And for 40 years, they failed. The hot desert climate, the humidity sealed inside ancient tombs, the chemicals used to preserve bodies for the afterlife, all of it seemed to destroy the one thing that could answer humanity's oldest question about Egypt's founders. Then, in a sterile laboratory in Germany, a team extracted something that should not have existed. Complete genetic sequences from bodies buried before the pyramids reached their peak. What they found overturned a century of assumptions. The founders of ancient Egypt were not who anyone expected. The question of where the ancient Egyptians came from has never been innocent. For over a century, scholars argued, some claimed the civilization must have been seeded by invaders from the Near East. Others insisted its roots were purely African. The debate became tangled with modern politics, colonial-era biases, and competing claims to ownership of the past. But without DNA, there was no way to settle it. The first serious attempt came in 1985. A young Swedish graduate student named Svante Pääbo, working secretly at night in his university laboratory, tried to extract genetic material from Egyptian mummy tissue. He claimed success. The scientific community was skeptical. Within years, those results were dismissed as contamination. Modern DNA had infiltrated the ancient samples. The hot Egyptian climate, it seemed, had simply destroyed any authentic genetic material thousands of years ago. By the early 2000s, the consensus was clear. Johannes Krauss, a geneticist at Germany's Max Planck Institute, would later describe it bluntly. Most experts had concluded that recovering reliable DNA from Egyptian mummies was essentially impossible. The heat, the humidity, the natron salts and tree resins used in mummification. Every factor seemed to conspire against preservation. DNA is hydrophilic. It absorbs water. And water, combined with heat and time, breaks the molecular chains into fragments too small and too damaged to read. For decades, this was scientific orthodoxy. Egypt would guard its genetic secrets. What changed was not in Egypt. It was in the technology. In the late 2000s, a revolution swept through genetics. High-throughput sequencing, machines capable of reading millions of DNA fragments simultaneously, replaced older methods that could only target specific genes. Instead of searching for a needle in a haystack, scientists could now sequence everything in a sample and sort it afterward. More critically, researchers had finally learned to recognize the fingerprint of authentic ancient DNA. When DNA degrades over thousands of years, it develops characteristic damage patterns. The chemical bonds at the ends of each fragment break down in predictable ways. Cytosine bases convert to uracil through a process called deamination. When sequencing machines read these damaged molecules, they appear as specific substitutions, cytosine to thymine, at the ends of strands. Modern contamination does not carry this signature. Only DNA that has been decaying for millennia does. This was the key, not just extracting DNA, but proving it was ancient. In 2017, Johannes Krauss and an international team turned their attention to a collection that had been sitting in German museums for over a century. The mummies came from Abusir el-Melek, a burial site along the Nile about 100 kilometers south of Cairo. The settlement had been devoted to Osiris, the god of the dead, making it a popular burial ground for centuries. The remains had been excavated in the early 1900s and shipped to the University of Tübingen and Berlin's Museum of Prehistory. 151 mummy heads. Ordinary Egyptians, not pharaohs, not nobility. Middle-class citizens buried in painted wooden coffins spanning 1,300 years of history, from the height of the New Kingdom through the Roman occupation. Krause's team tried something different. Instead of sampling the preserved soft tissue, the skin, muscle, and organs that earlier researchers had focused on, they went deeper. Into the bones, into the teeth. These dense structures, protected from humidity, had shielded their DNA from the worst degradation. The results stunned them. From 90 of those mummies, the team recovered complete mitochondrial genomes. From 3 individuals, they extracted genome-wide nuclear DNA, the full genetic blueprint passed down from both parents. Radiocarbon dating placed the oldest sample at 1388 BCE, the youngest dated to 426 CE. Between them lay the conquests of Assyria, Nubia, Persia, Greece, and Rome. Yet the genetic profiles barely changed. Across 13 centuries of foreign rule, the population of Abusir el-Melek remained remarkably stable. The DNA told a story of continuity, not replacement. But the truly unexpected finding was not about stability. It was about origin. When Krause's team compared the ancient Egyptian genomes to populations from around the world, both ancient and modern, a clear pattern emerged. The closest genetic matches were not in sub-Saharan Africa. They were in the Near East. The ancient inhabitants of Abusir el-Melek clustered with Neolithic and Bronze Age populations from the Levant, modern-day Israel, Jordan, Lebanon, Syria. They showed strong affinity with ancient Anatolians from what is now Turkey. Their Y chromosome haplogroups included markers common in the Middle East. The implications rippled through the field. Modern Egyptians, by contrast, carried significantly more sub-Saharan African ancestry, approximately 8 to 15% more than their ancient predecessors. This influx, the data suggested, occurred within the last 1,500 to 2,000 years, long after the Pharaonic era had ended. The ancient Egyptians, it appeared, were genetically closer to ancient peoples of the Eastern Mediterranean than to populations deeper in Africa. The 2017 study had limitations. All samples came from one site in Middle Egypt. The oldest dated to the New Kingdom, millennia after the pyramids were built and Egypt was unified. Critics argued the findings might not represent earlier populations or those from Upper Egypt, closer to Nubia. 8 years later, that gap began to close. In July 2025, a team from the Francis Crick Institute and Liverpool John Moores University announced they had done what no one had achieved before. They had sequenced the complete genome of an individual who lived during the Old Kingdom, the age when the Great Pyramids at Giza were constructed. The man had been buried at Nuwayrat, a village 265 kilometers south of Cairo. His body was discovered in 1902, interred inside a sealed ceramic pot within a rock-cut tomb. The pot and the hillside together had created a microenvironment stable enough to preserve his DNA for nearly 5,000 years. He was not mummified. Artificial preservation had not yet become standard practice. That, ironically, may have helped. Using shotgun sequencing on DNA extracted from the cementum of his teeth, the researchers built a genome at 2 times coverage, enough to map ancestry with confidence. The results echoed and extended the earlier findings. Approximately 80% of the Nuwayrat man's ancestry traced to ancient North African populations. The remaining 20% connected to something unexpected. Genomes from the Eastern Fertile Crescent, from Mesopotamia, from what is now Iraq. Not the Levant. Mesopotamia. The genetic modeling was precise. The best-fit statistical model showed 77.6% ancestry matching Middle Neolithic populations from Morocco, themselves a blend of Levantine farmers and earlier North African groups. The remaining 22.4% aligned most closely with Neolithic Mesopotamian genomes dated to 9,000 to 8,000 BCE. The connection was not a secondary effect. The statistical tests showed direct affinity with Mesopotamia itself. This suggests migration flows from the Eastern Fertile Crescent into Egypt during or before the early dynastic period. The routes remain debated, possibly along Mediterranean sea lanes, possibly through the Red Sea corridor, possibly through intermediary populations in the Levant that have not yet been sampled. But the genetic evidence now confirmed what archaeologists had long suspected from cultural artifacts: pottery styles, cylinder seals, architectural motifs that seem to echo Mesopotamian originals appearing in pre-dynastic Egypt. The exchange was not merely objects and ideas, it was people. The Nuwayrat man lived during a pivotal era. Radiocarbon dating placed him between 2855 BCE and 2570 BCE, overlapping the transition from the early dynastic period into the Old Kingdom. The first pyramids were rising. Upper and Lower Egypt had recently unified. The foundations of one of history's longest-lasting civilizations were being laid. His skeleton told its own story. Bioarchaeologist Joel Irish estimated his age at death between 44 and 64 years, extraordinary old for that period, equivalent to reaching one's 80s today. His bones showed extensive osteoarthritis and muscle development consistent with decades of hard physical labor. His height was modest, 157 to 160 centimeters. Genetic analysis predicted brown eyes, brown hair, and dark skin, common features among present-day Egyptians and North Africans. He was born in Egypt. Isotope signatures in his teeth matched the diet and climate of the Nile Valley. This was no foreign merchant or diplomat buried far from home. This was an Egyptian. His genes, however, told a story that reached far beyond the Nile. These findings do not settle every question. They sharpen some and open others. The Nuwayrat genome represents one individual from one site at one period. Egypt was vast. Upper Egypt, closer to Nubia, may show different patterns. The Delta region in the north, exposed to Mediterranean trade, likely absorbed different influences. A complete genetic history of Egypt will require dozens or hundreds of sequenced genomes spanning thousands of years and the full length of the Nile. Some scholars have questioned whether the Abuzer El Melek site, used in the 2017 study, may have drawn populations with atypical ancestry, possibly descendants of foreign mercenaries or settlers granted land in the Fayum region during the New Kingdom. Others argue the sampling remains too limited to draw sweeping conclusions about all ancient Egyptians, especially those from the earliest pre-dynastic periods when the foundations of Egyptian culture were being laid. The debate continues, as it should. What the DNA has established beyond reasonable doubt is this: the people who built and maintained Egyptian civilization across its first 3,000 years were not replaced by waves of conquerors. Their genetic profile remained stable through the reigns of foreign dynasties. Persians ruled, Greeks ruled, Romans ruled, but the population endured. The foundational ancestry of these ancient Egyptians was a blend, predominantly North African, with deep roots in the populations that had occupied the Nile Valley and the Sahara when the desert was still green, but also carrying a significant component from West Asia, specifically from Mesopotamia. This mixing likely occurred before or during the very earliest phases of Egyptian state formation. It was not a later import. It was present at the foundation. Modern Egyptians, the data confirms, retain approximately 80 to 90% of this ancient genetic signature. The changes that occurred, the additional sub-Saharan African ancestry, came later, within the last 2 millennia. The continuity is more striking than the change. 40 years ago, Svante Pääbo stared at fragments from mummy tissue and dreamed of reading the genetic story of ancient Egypt. His early results were dismissed. The climate, the chemistry, the sheer passage of time seemed to have erased the record forever. Today, Pääbo holds a Nobel Prize for sequencing the Neanderthal genome, and the mummies he once studied have finally yielded their secrets to the next generation of technology he helped inspire. In a laboratory in Germany, in a museum in Liverpool, in the tooth of a man who labored through decades under the Egyptian sun nearly 5,000 years ago, the code survives. It tells a story more complex than any single narrative about race or origin, a story of populations moving, mixing, building, then enduring across millennia while empires rose and fell around them. The secret buried in the tombs was never about who the Egyptians were. It was about how long they remained. What do you think? Leave a comment below. Don't forget to subscribe for more.